Hello, in this video we will discuss about the liver physiology is the second video. So the previous video you can watch the liver physiology number 1. So here is the production of bile. So in this way the production of bile mean the produce bile from the gallbladder. So here is the pancreas, stomach and liver contain bile. So the in the form of bile acid. So here is the fat, different type of fat. For example, unsaturated fat, which that is a oil, while the saturated fat, which that is in the form of solid at room temperature. So this fat, when we will intake, go into the stomach and not digest into the stomach, but when go into the duodenum, so the digestion will begin. And how it's possible? Here is the CCK, which that is sense the fat and give the signal to the gallbladder to constrict the gallbladder. And after the constriction of the gallbladder walls to secrete the bile acid, and this bile acid move into the duodenum. And after this movement of that into the duodenum, also pancreas produce the amylase and amylase also, but here is in the case of fat, so the lipase, pancreatic lipase. So here is the red blood cell breakdown to produce bile pigment, and this bile pigment store into the gallbladder, and this bile production and bile pigment and bile acid. So the bile pigment is another thing, but why bile acid is the another. So the bile acid is the amphiphilic steroids. What that means? That means the water. Amphiphilic mean the amphiphilic mean the two sides. That is the amphiphilic mean two sides. The water. One side is used for the water dreading, and one side is the water loving and the water hating. So here is the cholic acid and trans cholic acid and deoxycholic acid, which that is produced, and that means the micelles emulsification of fat will be occur, and this emulsification is the inside is the basically is the lipophilic, while outside is the hydrophilic, and lipophilic and lipophobic part. So in this way the bile salt will be interact to form a emulsification. It means the salt will be attract here, you can see. And here is the synthesis of amino acid also occur in the liver. So let's begin to understand the liver synthesis of the amino acid. So here is the synthesis of amino acids in the form of transamination and deamination. So after the deamination to produce ammonia, and ammonia convert into the urea, which that is the previous video whole detail about that. So here is the alpha keto acid, which that is add the ammonia and to become a alpha amino acid. So this is the ammonia. While on the other hand, the alpha glutamic acid. This alpha ketoglutamic acid is basically is the ammonia, which that is donation. So that basically is the donation to the another amino acid to the pyruvic acid, and it means the pyruvic acid is to neoaminogenesis will be occur, and the alanine formation, and this is the alpha ketoglutamic acid form donation, alpha ketoglutamic acid to form alanine. So this is the basically is the another production, basically is the formation of amino acid. So it means the synthesis of protein also, the amino acid and albumin. So here is the oxidative deamination will be occur in this way. Here is the L-glutamic acid, which that is dehydrogenation to form NADH, and this will convert into the water hydration to form a separation of the ammonia. So in this mechanism, the separation of the ammonia is known as the oxidative deamination. Why oxidative? Because the water will be added. It means the water contain oxygen. So here is synthesis of protein. We will discuss first is the clotting factor and anticoagulants, fibrinogen and antithrombin and alpha 2 microglobulin. This is the clotting factor which that is used for the blood clotting, and albumin is 50% of total protein synthesis is for maintenance of osmolarity and the transport of various hormones. So it means the albumin is the 50%, and in our body basically the water retention, sodium retention, as well as other the blood pressure regulation is due to the albumin. While on the other hand, here is the synthesis of protein to produce C-reactive protein. This C-reactive protein is reacted with others molecule uses to catabolism, and it is used for the catabolism. And on the other hand, the opsonin for use for the opsonization of pathogen and degradation and complement protein that work as defense. So C1 to C9 type of protein. On the other hand, the insulin-like growth factor, and this growth and development is due to the insulin-like growth factor, and it is the glucose absorption through insulin-like growth factor and the thrombopoietin production of platelets and angiotensinogen. So the IGF is produced due to the growth hormone production from the pituitary somatostatin, and it means the IGF will be produced. It means the insulin alternate path. So here is the transferrin, iron containing transferrin carry in protein, and IGF is a binding protein and carrier IGF-1, and it is used for the growth and development. So the transferrin is used for the storage of iron, and on the other hand, the other major protein also for use as a storage. So this was the storage about that. So here is the amino acid we will discuss here. If so, the amino acid also convert into the pyruvate, that is the catabolism, while the amino acid is converted into the ammonia, the separation of the nitrogenous part. So this ammonia will bind with the carbon dioxide as well as the ornithine to convert citrulline, and again second ammonia will be absorbed and arginosuccinate formation and this arginine formation. So this is the basic mechanism in this the liver, that is a cycle is occurring. And on the other hand, synthesis of a plasma protein, albumin and transferrin used for the storage of the iron is the transferrin. So here is urea production. After the arginosuccinate to arginine to produce urea, this urea will be excreted from the kidney. So here you can see in this diagram. So on the other hand, important thing is that the carbohydrate metabolism also occur. So the carbohydrate metabolism means the conversion of the glucose into the glycogen as well as the conversion of glycogen into the glucose. That is known as the basically gluconeogenesis. Genesis, and the basically you can see here is the glycogenesis. So the gluconeogenesis is the basically the formation of glucose from the pyruvate or other things, for example the lactic acid, while on the other hand the glycogenesis is the formation of the glycogen, and this is the phosphorylation and dephosphorylation will begin to produce the glycolysis and separate to produce pyruvate, acetyl-CoA and lactic acid. So this is the catabolism. So next here is the production of bilirubin metabolism we will discuss. The previous video was the overview. Here we will discuss about the production of bilirubin with the metabolism. So we will discuss here is the hepatocyte, and that is go into the unconjugated bilirubin formation. Let's begin to understand first of all. Here is a blood vessel. This blood vessel is basically the extra and intravascular hemolysis of red blood cell will be occur into the spleen and liver. After this, the unconjugated bilirubin will be separated, and this go into the blood circulatory system, and we may remain in the blood circulatory system, and that is bind with albumin. So this unconjugated bilirubin will bind with albumin, and albumin are present in the circulatory system. So after the binding with the circulatory system with the albumin, so it will be moved toward the liver. So remember about that. And after the movement into the liver, so albumin will be bind also here is, and it will become unconjugated to the conjugated bilirubin because albumin will be bind. So you can see that it is not occur in the circulatory system but into the liver. So after this, conjugation will move and store into the gallbladder and excrete into the large intestine. First small intestine and then large intestine. So in the large intestine, the conjugated bilirubin will once again convert into the conjugated to the unconjugated. So the bacterial proteolysis will be occur, which that is convert the conjugated bilirubin into the unconjugated bilirubin once again. And this unconjugated bilirubin is the water soluble. Basically is not water soluble, fat soluble, but the conjugated bilirubin is the water soluble. So in this way, the here is a bacterial proteolysis will be occur due to the microbiome are present in the large intestine. So in this way, the 90% bilirubin will be excreted through feces, and the fecal matter will contain 90% bilirubin as compared to that. So in this way, the 100% to 90% will be excreted from feces, while the 10% is left behind. 10% remaining, the remnant convert into the urobinogen, and the urobinogen is also excreted through the bacterial proteolysis to convert the conjugated bilirubin to the urobinogen. Urobinogen is the unconjugated. So in this way, the go into the 10% into the blood is the urobinogen. The urobinogen is go again into once again go into the liver, and after go into the liver to again that is also move toward the circulatory system and excrete through excretory unit of the nephron from the kidney. So that's urine is contain 10% remnant urobinogen. So this is the urobinogen is in the formation. So thanks for watching.